Hello, everyone, and welcome to our Global Health Tuesday webinar with Dr. Peter Halfmann. This seminar series hosts researchers and practitioners from the University of Wisconsin and around the world. Our speakers showcase the complexity of global health challenges and the many kinds of expertise needed to address them. By sharing their experiences and knowledge, these presenters provide insights into global health and encourage conversation to connect colleagues locally and globally. A couple of housekeeping items. You are automatically muted as you join the seminar. If you have questions for Dr. Halfmann, please put them in the chat box, not the question and answer, but the chat box, and we'll have time for questions at the conclusion of his presentation. One more item before I introduce Dr. Halfmann. I want to give you a heads up about this year's annual Global Health Symposium. Symposium will be held virtually, of course, on April 14th, 2021. Our theme will be Building Resilience Through Scientific Knowledge and Indigenous Wisdom, and we hope to see many of you as presenters or attendees. Please watch for more information about this in the coming weeks. Now I'm pleased to introduce Dr. Peter Halfmann, who will present From the Ebola Outbreak to the COVID-19 Pandemic. Virus Research Continues. Dr. Halfmann is a tried and true Badger, earning his undergraduate and doctoral degrees from UW-Madison. He is currently Research Associate Professor at the Influenza Research Institute, which is part of the School of Veterinary Medicine on campus. He is also a visiting researcher at the National Institutes of Allergy and Infectious Diseases, Rocky Mountain Laboratory in Montana, a high biocontainment BSL-4 lab. Dr. Halfmann has worked for more than 20 years with Professor Yoshihiro Kawoka, and his research focuses on emerging and re-emerging viruses like Ebola virus and SARS-CoV-2. Among his many accomplishments, he has developed a whole virus vaccine against Ebola virus that is in phase 1 clinical trials in Japan. And during the West Africa Ebola outbreak, he established a research lab in Sierra Leone to examine the body's responses to Ebola virus infection. A seed grant from the Global Health Institute also allowed him to explore novel viruses and paths during his work in Sierra Leone. I've had the pleasure of knowing Peter for the past 20 years and am thrilled to have him as our presenter this evening. I ask you to please join me in welcoming him.
Thank you, Chris. First, I'd like to thank everyone. I appreciate you joining this online presentation. As you can imagine, my days are spent in the lab mainly, so presenting our data for the last couple years here is a nice change of pace. So the first part of my presentation is going to focus on our research in Sierra Leone. It will mainly focus on Ebola virus research that was taking place during the outbreak in West Africa. And then I'll transition into our research on other viral pathogens in the country. The second half of the presentation is going to focus on our current SARS-CoV-2 research. And I'll highlight and focus on the establishment of animal models to study this virus, and then the use of these animal models to examine virus transmission. So just to give a little history and background on Ebola virus, it is the causative agent of a hemorrhagic fever virus or hemorrhagic fever disease. The early symptoms of infection resemble those of numerous other infectious diseases in endemic areas, and they include fever, chills, and body aches. However, in the later stage of the disease, which is known as the wet stage of disease, the characteristic symptoms of the disease appear, and they include the bloody diarrhea, blood in the vomit, skin rash or petechia, and bleeding from the body, mainly from the gums. Individuals who enter this late stage of the disease are often less likely to survive, and the overall case fatality rates can range from 40% to 90% depending on the outbreak. Now, Ebola virus is a zoonotic disease. The initial transmission into the human population occurs during the handling of infected animals. This is either by the butchering or the consumption of raw meat. And these animals often include bats that really show no signs of disease, or non-human primates such as chimpanzees. Now, once in the human population, the virus can transmit by contact with virus-laden fluids. Only 1 ml of fluid from an infected individual can be teeming with up to 100 million virus particles. So you can imagine during this wet stage of disease, a person is highly contagious. Now, experimentally in animal models, it only takes 1 infectious virus particle to cause a lethal infection. So posters were often distributed throughout Sierra Leone, warning people not to touch dead bodies and to avoid casual contact. So Ebola virus, which was always called Zaire Ebola virus, is a member of a much larger family of viruses called filoviruses. They are antigenically different from each other. So when you are talking about making a vaccine against one virus, it won't have cross protection against other viruses in the family. Some of the newest members of the family that include Bambali, Lovu, and Mengele virus have only been identified through sequencing efforts in bats. And no live virus has been identified, and no known human infections have been known to occur. So in this timeline, you can see filovirus outbreaks that have occurred. And 2 things just to point out, that these outbreaks have taken place in Central Africa, and that the number infected have ranged from only a couple individuals to a few hundred. Now, all of this changed in 2013 with the West Africa outbreak. This was the first outbreak in West Africa, and this was also the largest outbreak. It was 11 times larger in the number of infected individuals compared to all the previous outbreaks combined. Again, the outbreak started in late 2013 and finally was declared over 2.5 years later in June of 2016. There were over 28,000 infected individuals, and this resulted in over 11,000 deaths. This had a case fatality roughly of 40%. However, during the height of the outbreak, it was very hard to keep track of the number infected and the number dead. So these numbers are probably underreported. Some of the major statistics was in these countries, there were already, before the outbreak, limited number of healthcare workers, doctors, and nurses. And this outbreak caused at least 500 deaths of healthcare workers. So this really put a hard hit on the healthcare system in this country, in these countries. And currently there's over 17,000 survivors of this Ebola outbreak, and many of these people still suffer the effects of the virus. The scientists were able to pinpoint that this disease or this outbreak originated in Guinea back in December of 2013. This was most likely by an interaction with a child in an infected bat. However, after that initial contact, the virus spread quietly without any kind of recognition for a good 3 months. That was before this bulletin was released in March of 2014. It was describing a new disease that had claimed already 8 lives and infected many other people in Guinea. The symptoms included bleeding, but since this area was really known for Lassa virus and Lassa virus fever, that's what the causative agent was believed to be. It was Lassa virus and not Ebola virus. However, a couple weeks later, reference labs in various parts of the world identified the causative agent here to be Zaire Ebola virus. Now, 1 month after that, of these initial reports of infections, the outbreak grew. Besides Guinea, it was affecting countries of Sierra Leone and Liberia, and these were the hardest hit countries during this outbreak. And then in 2014, which was nearly 1 year after that first infection of the child in Guinea, the virus was uncontrolled in these 3 countries and had spread domestically in Nigeria, Senegal, and had then spread also internationally to Spain and to the United States. So it was around this point that Yoshi was introduced to Al-Haji here pictured. Al-Haji actually was from Sierra Leone. He was born and raised there, came over to the United States after the Civil War. And at the time that we met Al-Haji, he was actually had a position here at the School of Veterinary Medicine on campus. And he also was a professor at the University of Sierra Leone. So we asked him to come out to Research Park here at the IRI and kind of discuss what we wanted to do in Sierra Leone and see how possible that was. After a really productive meeting with him, we made arrangements to travel to Sierra Leone then, right before Christmas, to try to organize a research lab in the country. The goals of this research lab in the short term were we wanted to try to gain a better understanding into the insights of the mechanism that Ebola virus causes in humans. And already then we wanted to make a commitment early on to be in the country even after this outbreak. In doing so, we kind of wanted to expand on virus surveillance activities, look for new viruses in that actually region that were important for humans and animals. In doing so, we hope to prepare for future outbreaks and, more importantly, kind of raise awareness of infectious diseases in these areas. As you can imagine, this research team of investigators and collaborators was quite large. A lot of this really had to do with the approval of the government of Sierra Leone. When we landed and got to Sierra Leone in our first trip in December, we visited many hospitals, such as the government hospital in Lumley. We also visited labs and clinics, try to get a sense of the resources they had at the time, what their Ebola virus caseload looked like, and kind of get a better idea of what their workflow looked like. One of the most productive meetings that we had kind of near the end of this trip was with this gentleman, Dr. Fode Sar. At the time, he was the commanding officer at 34 Military Hospital. And given his role during the outbreak, he became the first Surgeon General of the country after the outbreak, and now is the Vice Chancellor of the University of Sierra Leone. Dr. Fode Sar managed a team of 12 doctors and nurses and lab technicians at this Ebola treatment unit at 34 Military Hospital. And though it was a small ETU with only 8 beds that they had in the red zone, this particular ETU was really known for having a high survival rate than other Ebola treatment centers in the area. So he got a lot of people coming to him asking for treatment during this outbreak. Now, a short distance from the Ebola treatment unit was a building. Inside this building, there was 4 rooms, and one of these rooms shown here was actually empty at the time that we visited. So Fode Sar was generous enough to give us this room to set up our research lab. With the number of canceled flights, especially cargo or freight flights that were going into the affected countries, it was really difficult to get supplies, reagents, and equipment in. What we really had to do was bring all these items with us in suitcases and in boxes on the flights that we were on. The other big challenge was virus containment. At the time of the outbreak, I had roughly 10 years of experience working with Ebola virus. But this was all in controlled, high containment biosafety level 4 labs. So to start working with Ebola virus outside of these labs, safety was kind of a major concern of ours. So to try to tackle virus containment and keeping the researchers safe while we're working with blood samples that have a lot of Ebola virus in it, we brought these glove boxes, or these tents. These glove boxes zipped up, were airtight. There was a blower to create negative pressure inside the tent, and then all that air was then HEPA filtered exhaust on its way out. During our downtime, and since these tents were new to us, we had spent a lot of time practicing with non-infectious samples, trying to develop protocols for inactivation of the samples that we had, and trying to figure out workflows for moving from one tent to another tent. This way we could safely work with blood samples that contained the virus. Now, previously our work with Ebola virus was all done in animal models. We looked at the host response in mice with mouse-adapted virus or with wild-type virus in non-human primates. But in both of these situations, the virus was always 100% lethal. We only had one type of sample that we could collect from these animals. But now during this outbreak, we would have access to human samples. And more importantly, we would have access to samples with two different kinds of disease outcomes. And we had samples from individuals that got sick, their body was unable to mount an appropriate immune response to the virus, and the virus would win and overcome them. So we had these fatal samples. And then we also had situations where individuals became infected, but their body was able to mount an appropriate immune response. They were able to suppress virus replication and then survive infection. So the aim of this first study was to really examine the global changes that were occurring within the human body during Ebola virus infection, using these two different kinds of outcomes of infection. This involved a very large team of researchers, both from the University of Wisconsin and the University of Tokyo, and then clinical staff and clinical technicians in Sierra Leone played a huge support role in this. We always wanted to have 2 researchers in the lab, sometimes 3 researchers at the height of the outbreak, to safely process samples and to kind of have a buddy system to make sure nothing went wrong. I mentioned that Fode-Sar had an Ebola treatment center at 34 Military, and it was a small one. He also was able to gain samples from a much larger 50 to 60 bed treatment center in Hastings. And now from these 2 sites, we took kind of a global approach to look at how the humans responded to infection. We had cohorts, we had survivors, and from survivors, we were able to get multiple samples during the course of infection, and these samples were whole blood samples. We also had fatal cases. From fatal cases, we were only able to get 1 blood sample, and this was kind of the initial blood sample at the time of diagnosis. And then we had a cohort of healthy individuals that never saw Ebola virus. Now we took these samples and we isolated the plasma and that buffy coat that contains all the blood cells. We did this by centrifugation over a Phi-Col gradient. And then we performed what is known as omics, or the ability to look at changes in proteins, lipids, metabolites in the plasma. And then we also looked at RNA changes or transcriptomics going on in those blood cells that we obtained after centrifugation. Then some other standard assays that we performed were looking at viral gene copies in the blood cells, and then also looking at inflammatory cytokines in the plasma. So one of the first assays we performed, and we performed this in Sierra Leone because we were using serum samples with live virus in them, were these ELISAs to look at cytokines in the plasma. Inflammatory cytokines such as TNF-alpha and IL-16 were highly upregulated in people with fatal cases of Ebola virus infection and fairly low concentrations in the survivors. And of course, this was very consistent with other studies. These inflammatory cytokines were studied for many years. However, it let us know that our samples, this data let us know our samples were actually in good condition, and we could move on to the more kind of omics approaches to look at what was going on. So a new piece of information that we found from the study was that the involvement of neutrophils in Ebola virus pathogenicity. The first set of data to indicate this was from the RNA-seq data. And the RNA, again, was obtained from these blood cells collected during centrifugation over Phi-Col. Now, normally, neutrophils would not be part of this buffy coat. They would actually be in the bottom layer and not associated with the buffy coat at all. However, neutrophils will fractionate with the blood cells when they're abnormal in such diseases as sepsis. So finding these neutrophils in the blood fraction gave a good indication that we had some quite abnormal neutrophils from these fatalities. And you can see here that we have many markers for neutrophils in this blood sample shown here. And they were significantly, the expression of these markers were significantly increased in the fatalities versus the multiple survival samples that we got. We also saw an involvement for neutrophil and Ebola virus pathogenicity in the proteomics data from the plasma. We saw proteins that were associated with these neutrophil extracellular traps. They were highly expressed in the plasma of people that succumbed to infection with Ebola virus, but we didn't see these in the healthy individuals. And as you can see here, we had significant increase in expression in these three markers that signify these nets. So also using these samples, we also were interested in identifying biomarkers that could differentiate the course of disease outcome. We wanted to look at biomarkers. We had particular candidates from the plasma. This included lipids, metabolites, cytokines, some other miscellaneous proteins, and we used the virus genome as kind of a gold standard. So what you can see here on the left-hand side is the viral genome copy in the plasma. This is used to kind of tell apart fatality cases, and they're pretty consistent over time with the fatalities. The number of viral gene copies stays the same. But what you can see with the survivors, they're kind of all over the place. So we found two molecules, L-threonine and vitamin D binding protein shown here, that really stratify patients better than this gold standard of the viral genome copies. And you can see they kind of segregate from each other, survivors here, fatality cases, and more importantly, they're really stable over time for the survivors. So from the first study, we can kind of conclude that neutrophils play a role in Ebola virus pathogenicity. We also saw that pancreatic enzymes were highly expressed in fatality cases, something I didn't get a chance to talk about, but these two new findings could lead to the development of more targeted drugs that could prevent the fatality outcome of disease. We also identified biomarkers for the severe cases of Ebola virus infection. We hope to use these to try to improve diagnosis and hopefully try to aid those in how we prioritize the treatments and resources that are quite limited in outbreaks. So education is a huge part of getting community involvement into the viruses and the diseases that they kind of bring. There was a lot of billboards up in Sierra Leone letting people know that Ebola virus is actually real. So after the outbreak, we kind of want to continue this education of infectious diseases in Sierra Leone. And we did this in a couple different ways. In McKinney, which is about 2 or 3 hours from Freetown, where we kind of were stationed, we held these health fairs to kind of promote health awareness. And these were very simple kind of health measurements. It wasn't anything major, but the biggest thing was try to get the community out and to engage with the researchers there and to try to get them to start thinking about healthy eating habits, exercise, waste disposal, and infectious diseases, along with hygiene. So we held these fairs. We gathered a bunch of kind of information such as height or blood pressure. We took people's pictures and kind of gave them out as a gift at the end. So this took part during the day. And then in collaboration with Sony out of Japan, we kind of held a nighttime movie that actually attracted a large population of the town that we were in. And then halfway through these movies, we would share educational commercials. These commercials were directed by local groups, and they also had actors that were locals too. And it kind of, again, reiterated good hygiene, good eating habits, and kind of reminded people of the town about infectious diseases such as Ebola virus and other viral diseases out there. Now, it was through this kind of outreach program with the community that we became involved with the Sierra Leone Association of Ebola Survivors. They kind of contacted us wanting to know how we could help them out. And what they were experiencing after they survived Ebola virus infection was kind of some discrimination as Ebola virus survivors. So we kind of hosted training seminars for them for becoming paramedics, taxi cab drivers, and other kind of careers that could help them gain a footing in now post-Ebola Sierra Leone. And in talking with them, they were interested in knowing their antibody status now after that they survived Ebola virus infection. And we were kind of interested in knowing the subclinical cases of close contacts that they had in their household. So with this association, we kind of did a study on looking at samples obtained from Ebola virus survivors 6 to 18 months after they had infection. So we had an educational day to talk about antibody responses and what we would do in this study. And then volunteers donated blood from themselves, and then they also recruited family members. And we also did some recruiting with close contacts, such as healthcare workers. And these blood samples were collected at a survivor clinic. And then we transported these samples to our lab in Freetown for processing. And what we wanted to know was to look at the antibody levels, the IgG levels, against 3 major proteins of Ebola virus 6 to 8 months or 18 months now after infection to see what they look like. We looked at antibodies against the glycoprotein, the surface protein of the virus, NP, which is the major protein of the virus that forms kind of a coat around the RNA, and then VP40, which is another coat protein that kind of encapsulates the virus. So what you can see here in the survivors, we have a wide range, but of a very robust antibody response to all 3 of these viral proteins. And interesting, we started seeing in some of these close contacts, again, these were family members that took care of the sick or healthcare workers. We saw antibody responses to some of these viral proteins too. Of course, not in the same magnitude or percentage as the survivors, but they were there. So this table just kind of shows that we looked at 240 total survivors. We had antibody responses to all three major antigens in 84% of them. In 5 individuals, we didn't detect any antibody response, but this very well could be due to some clerical issues with recording of all of these samples that we got. We also had 264 or 68 close contacts, and this was divided up into 57 healthcare workers, and a large number, 211, were relatives that took care of the sick. And what you can see is that we did have people that had antibody responses to infection. So what we kind of wanted to do was just to verify these antibody results by ELISA by looking at neutralization titers. And here on the top are close contacts. And which was surprising was that we did have at least 12% of the close contacts did have neutralizing antibodies against Ebola virus. And this indicates that there was potentially some kind of subclinical infection, perhaps while they were taking care of a relative. And of course, this has been published in previous outbreaks and has been now published after our publication here. So again, now that we're in post-Ebola Sierra Leone, our group still wanted to remain engaged in research. So we kind of started research on vector-borne viruses in the country. Now, there was some previous work done on this by others, but their surveillance was quite limited. And this was a few years back, so we kind of wanted to get an updated current assessment of some of these viral vector viruses. One of the major issues or obstacles in Sierra Leone is the lack of diagnostic capacity. Talking with a lot of people at clinics, when you walk in with a fever or chills, you're often just given malaria medication without any kind of testing. So what we wanted to do was kind of form some new collaborations with clinics. We started a dialogue with technicians in the area to discuss what kind of techniques were well suited for the country. And we wanted to make sure that the protocols and the equipment that we were going to establish and get going were going to be suitable, because at this time, after the outbreak, a lot of the non-government organizations were leaving Sierra Leone and leaving behind them these expensive QRT PCR machines that were used for Ebola diagnostics, or now we're just collecting dust because they either were broken down and there was no service on them, or they couldn't get reagents. So making sure that we communicated with the people that were going to carry out these diagnostic assays and making sure that we're sustainable was really important to us. So the first step to kind of get this rolling was surveillance. Surveillance being a key step in the infectious disease prevention process. We wanted to evaluate the current prevalence of viral diseases by serological examination. So basically finding out what was circulating in Sierra Leone at the time. We decided to look at chikungunya virus, Zika virus, and West Nile virus, all viruses that cause fever and chills, and that were transmitted by mosquitoes. So now this collaboration, which was funded by some seed money through the Global Health Initiative, was at 34 Military Hospital with laboratory technicians. So we had established a good working rapport with that group there. And then we also branched out to Ramsey Medical Laboratory. This is Tadashi, who was a postdoc at the time in our lab. He spent many hours or many months there working with the technicians at Ramsey. Now they gathered serum samples that were from blood. These blood samples were going to originally be discarded. So we used them for this study. And these blood samples were collected from patients in Freetown from October 2017 to May of 2019. And in total, they were able to collect over 2,000 blood samples and obtain a nice aliquot of serum from them. So for the first type of diagnostic assay that we collaborated with was just a simple indirect IgG ELISA to look at past viral experiences. And for the technicians at Ramsey and at 34 Military, this was important for them because they would run such ELISA assays for HIV testing. So to increase their skills on this type of assay was important for them. And this was also a very simple colorimetric readout for a positive sample shown here. The plate readers were very portable, fairly cheap and kind of hooked up to their existing software and computers that they had at the labs. So these were the results for chikungunya virus. Patient samples that we got from Ramsey had about a 30% prevalence of previous infection with that virus. And at 34 Military Hospital, roughly 43%. So this was basically a total of 35% of the samples that we looked at were IgG positive for chikungunya. To kind of validate this ELISA that we were running, since this was a homemade ELISA, we also sent samples back to UW-Madison, and we did a neutralization assay looking for the ability of those antibodies to neutralize virus. And what you can see is that the results were quite similar. So this homemade ELISA that we developed with the people in Sierra Leone was fairly reliable. What was new here is that no one had really thought about chikungunya virus circulating in Freetown at all. So this is something that we discussed with the Ministry of Health and Sanitation. And right now we're kind of working out a way to differentiate chikungunya virus from some of the other alpha viruses in that family, kind of be able to have a differential ELISA to kind of tell the two different, or to tell the viruses in that family apart. We also looked at Zika and West Nile, and they weren't as prevalent, but we still were detecting roughly 8% of our samples were Zika positive against the NS1 antigen, and 3% of those samples were positive against the E antigen for West Nile virus. So now that we've kind of started establishing viruses in the environment, we want to look at zoonotic viruses in animals in Sierra Leone. This again was a big collaboration between UW, the University of Sierra Leone, and Hokkaido University. We first looked at ticks in cattle that were ready for market, that were going to go to Sierra Leone from outside in Waterloo. And you can see that these cattle were heavily infested with ticks. These ticks were collected and virus sequence analysis was performed. We also gathered ticks from flagging of vegetation, and we also collected mosquitoes. To highlight one of the important findings, though, is that from these ticks, we were able to identify virus from 3 of them, and one of these ticks actually had Rift Valley fever virus, which was not known to be in Sierra Leone or in Freetown area. So again, this was a new virus in the area that we reported to the Ministry of Health. We also looked at the virome of bats in Sierra Leone. We contacted rangers in the villagers of the area and looked at the Mambaya forest area. And this was important because previously the CDC announced that they found Marburg virus, which is related to Ebola virus, in this area. We did live trapping with nets, caught some insect-eating bats, and then took those samples for RNA sequencing, ran it through a battery of different algorithms to try to find out what kind of viruses or viruses that were close in homology. And we did not find any filoviruses, but we were able to find some retroviruses in these bat population and some pox viruses. So this was kind of our preliminary run, and we're kind of looking at these samples a little more. But now, right now, our research efforts are kind of on pause because of the current outbreak. So I kind of want to switch over in this last section here to talk about SARS-CoV-2. I remember getting the first ProMed email about this virus January 1st, which seems like a lifetime ago, talking about a mysterious virus pneumonia in China. It was only a week later that scientists were able to identify this outbreak and linked it to a novel coronavirus. This novel coronavirus in 2019 was later named SARS-CoV-2, and it's the infectious agent in humans of the disease COVID-19. In February 5th of 2020, Wisconsin had its first case, and this gave us access to our first clinical sample for research purposes. And since then, at the end of November here, we have over 3,000 virus isolates stored away in the freezer. This is thanks to Bill and Molly and the staff at the UW Hospital Clinical Diagnostic Lab. Early on in the outbreak, we knew that this was going to be a novel virus of potential, pandemic potential. So we knew that we needed to develop animal models to be able to evaluate the efficacy of vaccines and therapeutics, and also to look at transmission and viral pathogenicity. Now, what was important for this is that the animal had to recapitulate the disease of COVID-19 that is seen in humans. First, we infected wild-type mice, both 3 different strains, both young animals and older animals. But because the receptor ACE2 that the virus needs to get into cells isn't conducive for SARS-CoV-2 to enter, we never saw any kind of body weight loss in these animals. We then moved to transgenic mice that would express human ACE2. But the problem with human ACE2 transgenic mice is that these animals will also die from encephalitis, the infection of the brain. This is what we don't see in animals, or we don't see this in humans. So they don't make a really great model to study the virus. So then we turned to the primate center on campus and looked at marmosets. We looked at a wide range of animals from 1 year old to 13 years old. But after infection, even with a high dose of virus, we were never able to recover infectious virus from nasal swabs, oral swabs, or rectal swabs at various days after infection. So these animals were resistant to COVID-2 infection. So then we looked at Syrian hamsters. This is where we had our first success in the animal models. We looked at both young animals, which is about 1 month old, and older animals of 7 month old. These animals were infected with a low dose in blue or a higher dose of virus. And what you can first see here is that the animals that got the high dose of virus, the younger animals had about a 10% body weight loss, and the older animals had almost a 20% body weight loss. The virus grew very well in the lung tissue of these animals. You can see here on day 3 only that virus titers with a high dose of virus gave us 10 to the 8th PFU, or virus particles per gram, which is extremely high. Virus titers, the virus load, that reduced by 100-fold by day 6. We never saw virus in the kidney, the intestines, or in the blood. And these viral load data was very similar in young animals versus the old animals, high dose versus low dose. Next, we looked at pathology in these animals. These are younger hamsters that got a low dose. And what you can appreciate here is on day 6, there's a lot of infiltration of immune cells and a lot of inflammation. This is versus day 3. And then again, if you look on day 6 here, where we on the left with the high inflammation, this kind of disappears then on day 10 after infection. We next looked at CT scans of these hamsters that were infected. On the left-hand side here, you can see a CT scan of normal lung. It's quite clear. This is compared to a CT scan of a hamster that was infected 7 days after infection. You can notice the congestion going on, the pocket of air that's not supposed to be where it is. So these are very bad looking lungs. And then day 16 after infection, this is kind of cleared up. So hamsters that are infected with SARS-CoV-2 definitely share CT imaging characteristics that are very compatible, very similar to human COVID-19 disease, and making hamsters a very useful model to study this disease. This is just kind of summarizes the severity of disease. With both low and high dose infections, the severity kind of peaks at day 8 after infection. Virus titers shown here peak at day 3 after infection, come down by day 6, and then we don't see any virus on day 10 after infection. Now that we had a great model to study this virus, we wanted to look at the effects of a particular mutation that was catching the news, and this was a mutation in the spike protein at position 614. Early on in the pandemic, prior to March of 2020, you can see here in gold, which was the early virus, which had at position 614 the amino acid D. What you can kind of notice here in Europe, there was some blue virus, and this blue virus, which was a change at position 614 in the spike to glycine, kind of overtook the world by June of 2020. And what we wanted to know was, why did this happen? So to study this transmission of these two viruses, we collaborated with a researcher at the University of North Carolina, Ralph Barrick, and he has a technology to make infectious coronavirus through reverse genetics. So he made us 2 almost identical viruses, except they had 2 different amino acids at position 614. Gold is the asparic acid, and in blue is the glycine. So we had transmission cages set up for hamsters. We infected hamsters either with the early type of virus or the late type of virus. And then 1 day later, we put a naive hamster in the cage. Now, between these two hamsters was a divider of 15 centimeters. It kept the hamsters away from each other, so no direct contact, but allowed airflow between the two hamsters. So what we wanted to know was, what was the transmission kinetics here? If we looked at day 4 post-exposure, so day 4 after we introduced this naive animal, we were able to detect infectious virus in 8 out of 8 of hamsters in both of the virus groups. So there was no difference in transmission on day 4. But what about earlier on day 2? And this is where it was interesting. We didn't see any transmission with the earlier version of the virus, the 614D. However, in the virus that gained this glycine amino acid position 614, the virus that overtook the world, by June of 2020, we had transmission in 5 out of the 8 naive animals. So next we looked at a competition assay. We infected hamsters with equal amounts of both viruses, a 1-to-1 ratio, and then looked at what happened as we passaged the virus from hamster to hamster. And what you can see here in this cartoon is that the 614G virus overtook these hamsters, and that's all we found in the hamsters after 3 days, or after 3 passages. So this later virus that had the mutation in the 614 for G for the spike, it grew better and it transmitted better than this earlier version of virus. The other kind of headline that caught us was the transmission of coronavirus to pets, pet cats, and then the transmission of COVID-2 to larger animals at the Bronx Zoo. So we kind of wanted to look at transmission kinetics of the virus in domestic cats. So we infected 3 cats with the virus and then housed them for direct contact with naive animals. And we wanted to know, what did the transmission kinetics look like? So with these infected animals, we were able to detect virus from nasal swabs that we took out to day 5 and day 6 after infection. And then we were, no infectious virus was able to be detected. Interesting too that these cats that we infected actually had no symptoms of disease. We never saw any discharge from the nose, no coughing, no fever, and no body weight loss. Now with these naive cats that we placed 1 day after infection, we were able to detect virus as early as day 3 after infection and out to day 5 and 6. So the virus was easily transmissible to the co-house cat. And again, the virus then, we were not able to detect virus after 4 or 5 days then. So now this has implications in households that have cats and an infected person. This also has implications in cat shelters and veterinarian clinics. And we know that there's human-to-human transmission, human-to-cat transmission, and cat-to-cat transmission. What hasn't been reported was transmission from cats to human yet. However, this is not totally implausible. There's a lot of news out there about transmission within mink. Humans have transmitted the virus at mink ranches. Mink have transmitted the virus between them. And then there's growing evidence that mink can now have transmitted the virus back to humans. The same situation could happen in cats. So in summary, our COVID research has shown that hamsters are a very robust model for SARS-CoV-2 infection. Some research I haven't been able to talk about, but they've been very useful for the evaluation of novel vaccines and therapeutics. They're very useful in studying mutations that could cause attenuation, and they have given us better insights to look at transmission of virus. Just to wrap up, obviously this is again another large project that involved a lot of collaborators in Japan that helped us obtain patient samples, and a wide range of groups here at University of Wisconsin that helped carry out this research. Thank you for your attention. I'll take any questions from the audience now.
Thank you very much, Peter. Anne Renee, do we have questions in line in the chat?
No, we do.
Okay. Well, I'll take.
We'll wait for them.
take the prerogative of asking a question than myself. And actually a couple of questions about the both temporal and geographic distribution of Ebola. You know, when you showed your timeline of Ebola outbreaks, it's very obvious that there have been so many more outbreaks since the mid-1990s than there were previous to that time. And I'm curious what you think about that pattern. I guess in essence, do you think it's real? Is there really been that much more widespread distribution of the virus, or do you think it's an issue of recognition and diagnostics that have improved over time?
Yeah, so it's kind of a complicated question. What I didn't talk about was now after the West African outbreak, which was the largest outbreak, we experienced the second largest outbreak in the DRC. So since then, we've had two very large outbreaks in the Democratic Republic of the Congo. This probably has to do with the fact that we can recognize the virus a lot faster and diagnose it. But we also have a lot of going on with climate change and deforestation and people encroaching into the environment where bats and chimpanzees live that are harboring this virus. So it's multifold of what's probably going on. And also the fact that some of these outbreaks are not occurring in remote villages anymore, but they're occurring in kind of urban settings where there's a lot of transportation. And that kind of spreads the virus out quite fast now. And that's why we're probably seeing these larger outbreaks.
Great, thanks. Our good friend and colleague, Dr. Tom Ewell asked, oops, it moved, excuse me, asked, did your Sierra Leone tests also test for dengue?
Yeah, so we did get the 4 different kind of serotypes of dengue virus, and we're kind of working out the logistics to kind of distinguish between dengue virus 1, 2, 3, and 4, but it is something that we are looking at with using inactivated whole virus and then also some viral antigens to look at that. And we did have positive IgG samples for dengue virus.
You know, on a related question, again, our friend and colleague, Dr. Tony Goldberg asked, what's the specificity of your serologic assays for flaviviruses in Sierra Leone? With the low prevalences, could it be non-specific reactivity or reactivity to related viruses?
Yeah, so exactly right. With these serology tests, you're looking at potential cross-reactivity to other members in the family. So trying to find antigens that will give you real specific antigen results is needed. And also looking at more than one antigen is needed too to kind of start differentiating between all those different members of alpha viruses, flaviviruses, and even filoviruses.
Thanks. We have a question. Any therapeutics for COVID-19 tested in hamsters yet? And if so, any results you can talk about?
Yeah, so hamsters have been really hard to treat and offer protection against some of the drugs that are being talked about in the press. Again, hamsters there's no lethality in hamsters, so our readout are lung titers and body weight changes. But we don't have great reduction in virus titers in the lungs with the antivirus that we've tested. Maybe at best 1 log reduction. So when you're talking about 10 to the 8th BFU per gram of tissue going down to 10 to the 7th, that's still a lot of virus in the hamster lungs. So one avenue that we have gone down with a collaborator, Patrick Wilson at the University of Chicago, is monoclonal antibody therapy. And with that, when we kind of do a stringent model, infecting the hamsters first, and then the next day giving them a therapeutic dose of monoclonal antibody, we see a good 3 or 4 log reduction in virus titer, which is better than the chemical compounds we're looking at, but we haven't achieved no virus titer at all.
And Peter, Dr. Patz is asking, are there similarities between the receptors in cats and humans that are shared between those two species that are unique and not shared in other mammals? Any common denominators?
Yeah, so actually it's something that we're starting to tease apart. We cloned the marmoset ACE2. So marmosets, of course, didn't have an active infection. So we cloned that ACE2, and then we cloned the ACE2 of humans. And we've kind of pinpointed 3 critical amino acids in the ACE2 that we can mutate from marmoset to human forms. And then that gives us increased infectivity using that marmoset ACE2 now with human amino acids. And I can imagine in the cat probably shares these critical human amino acids too that allow them to become infected.
Along that same line, I'm curious about the marmoset studies that you did early on with COVID or SARS-CoV-2, and you didn't have evidence of viral replication and shedding by day 4 post-infection. Did you by any chance follow them for a longer period of time and see whether they seroconverted, even though they didn't appear to shed virus?
Yeah, we did. The issue with marmosets was the secondary antibody to detect the IgGs. We were able to find one. The antibody seems to react. It gives off a color change, but we weren't able to detect any SARS-CoV-2 specific antibodies or IgG antibodies. So it seemed like they never got the virus, even though we gave them a pretty high dose of virus, which was actually surprising.
And Dr. Yule said there have been some instances of SARS-CoV-2 viruses isolated from dogs. Will you be going to the dogs? Dr. Yule's humor as usual, but any plans for doing experimental studies in dogs?
No, there's been other groups that have done dogs. There's not much to gain from it. We are working with some great groups to get serum samples from dogs that they thought the owners were infected. They knew the owners were infected to see if the dogs had seroconverted. So kind of looking at that in-house transmission with household pets.
And I'm curious, have you gotten involved at all with the mink outbreak in Wisconsin?
Actually, no, we haven't. So it'd be interesting to see, and I know groups like Dave O'Connor and Tom's group will probably start sequencing that virus that's coming out of mink. So it'd be interesting to see if the minks in Wisconsin are picking up mutations that the mink in the Netherlands are picking up.
Or obviously, what they're getting infected with are the human viruses that are co-localized to that area of the state.
Yeah. So sequence analysis of looking at the transmission chains is going to be interesting.
Dr. Patz asked on a similar note, what's the likelihood of SARS-CoV-2 setting up in a North American reservoir host?
Yeah, so actually there was some studies that people have done infections of deer mice, and I believe deer mice can support virus infection. I don't think there's enough research, at least I don't know the research that's going on of looking at potential reservoirs in the wild that could be hosting this virus. And one of the greatest reservoirs of this virus could be humans. If we start getting a vaccine and the virus starts drifting away from that vaccine, we could start having seasonal, we don't want this, but we could start having seasonal effects of SARS-CoV-2 just like we do with flu. So we could be acting as the reservoir ourselves.
Right. Great. Well, I think that's the end of our questions in the chat. And I want to thank everyone for joining on the webinar this evening. We had a great turnout. And of course, I want to thank all the folks in the Global Health Institute that were so critical to putting this all together for us. So thanks to Logan, to Anne, to Monet, and others. We appreciate your efforts in pulling this all together. And of course, most importantly, we thank Dr. Halfmann for his time and knowledge and sharing that with all of us. So Peter, thank you very much. And to everyone, have a good rest of your evening. Stay safe, stay healthy, wear your mask and wash your hands. Thanks very much. Take care.
Thank you.